So I'm happy to introduce my co-chair, Anja Kipar, who will talk to us about SARS-CoV-2.
Okay, thank you. So this talk is about the effect or the mechanism of effect for monoclonal antibody treatment post-exposure with SARS-CoV-2. And this is work we've done, or it's building up on work we've done before, where we used Ronapreve, a mixture of 2 monoclonal antibodies. And you know this was conditionally approved for COVID-19 pre- and post-exposure prophylaxis and treatment. And we used that in the K18 mouse model and SARS-CoV-2 Delta, and we could show that when we used it post-challenge as a treatment, we significantly reduced the viral burden in the nasal turbinates, in the lungs, and also seemed to be able to block brain infection in the mice. So if you have a look at the weight curve, then there was no loss of body weight with the Ronapreve treatment, and there was significantly lower viral RNA loads at day 4 in the nasal turbinates, at day 6 in the lungs, and then we looked at it later, day 9, then we could see in this group of animals there was basically no viral RNA in the brain, and lung levels were significantly lower. So now with Omicron and so on, the sublineages, there is an efficacy loss. But we were actually interested in understanding the mechanisms that underpin the observations that we made, and also potentially inform the development of monoclonal antibodies for other viruses, and particularly pulmonary viruses. So question is, how does Ronapreve block brain infection? So we know that intranasal challenge with pathogenic isolates, if I summarize them like that, frequently leads to infection of the brain, and it starts from day 3 onwards. And it's not due to an overexpression of ACE2 protein in the mouse brain of human ACE2. That is not observed. But what is very likely is that the virus gets into the brain via the olfactory epithelium, olfactory nerve, and then olfactory bulb, and spreads very rapidly. And as we say, we still don't know why it likes to move from neuron to neuron, because it's most likely neural cell-to-cell spread. It induces in the mice, when it's widespread, the infection, a mild non-suppurative meningoencephalitis. And we can boil it down to, we need at least 10 to the 9 copies of N1 to see viral antigen expressed. So there's really quite high levels. But the question is, does Ronapreve block brain infection by reducing the viral burden in the nasal mucosa, or would it actually work directly in the brain? So we used the same study design as before. We looked at day 7 post-infection, infected, of course, as I said, with Delta, and then 24 hours after infection, the animals received IP Ronapreve, meaning the lungs were infected by that time, and there was sustainable nasal infection. So, and then we wanted to see the effect of Ronapreve, and that's like in this little scheme here. We want to see the effect of Ronapreve in infected animals, but also the effect of Ronapreve in healthy animals, and then see how it affects then the effect of Delta itself. So when you look at the infection, I want to draw your attention to Ronapreve treated mice only. There is nothing happening. No substantial weight loss over the study period. So it doesn't have a clinical effect. So, and then really it blocks brain infection insofar as we cannot detect viral antigen. There is no inflammatory response, nothing. And when you look at the viral RNA levels, they're just above the level of detection. So in the lungs, again, we could show it is substantially reduced, but day 7 now, and not earlier time points. When we look at these animals doing ELISA for the pharmacokinetics and the distribution of the antibodies, yeah, there are in serum and tissues a bit more the imdevimab than casirivimab, but I think this is not of major relevance. And it seems to be at a high level. But when you look at it, it's a low tissue to serum ratios. And it's even in the nasal turbinates, it doesn't go more than 1% of serum level. In the brain, it's particularly low, also 1%, a bit higher, of course, in lung and spleen and liver. Why do I say higher? Because we should look at the next slide. So, but also to show you, infection doesn't make any difference with regards to the distribution of the monoclonals. And so I said, of course, it makes sense. You have higher levels in, for example, the lung and the liver than you have in the brain. That's because we can only find the antibodies within the vessels. So it's in the serum. And this is a treated animal and uninfected. There is no evidence that it actually reaches the brain parenchyma. And when we then looked at the infected animals, the same is true. The only evidence of really what you'd call tissue penetration is in the lung here, and I had to choose a high magnification. There's a little bit in alveolar macrophages. So obviously with in the very thin-walled vessels in the lung and generally larger molecules can penetrate to some extent, it gets to the alveoli where it probably binds the virus directly. But in the brain, there is no evidence that they ever left the vessels. So we then were interested in looking at the transcriptome. That's part of a larger study we're doing. And when you look at the delta-infected animals, then really the ones that have viral antigen, they cluster together. And all others, let them be infected, treated, or untreated, they cluster together otherwise. So, but then let's have a bit of a closer look and compare the delta infected ones and to the mock infected, so untreated, and then delta infected to the delta infected Ronapreve treated ones. You can see that the volcano plots look pretty similar. So that would indicate that the Ronapreve treatment of the delta infected mimics these mock infected mice. So we had a bit of a closer look. What about the positively or negatively enriched pathways? Basically, it's really the same. It's the response to virus infection. It's innate immune response. But, and I try to highlight that with these arrows here, there are slight differences between the groups. So something's happening that doesn't make the delta infected Ronapreve treated animals mock infected. So when you then looked at it in the Venn diagram, you can see that there is a proportion of genes upregulated in both groups, but most is actually only SARS-CoV-2 delta, no treatment, and there is a proportion here that seems to be specific when they are Ronapreve treated. So we had a bit more of a closer look. When you look at the mock infected Ronapreve treated versus mock infected untreated animals, you want to see what Ronapreve does. And there are a few genes that are significantly infected, they're upregulated, and the question, of course, is then, is it due to the monoclonals in the serum? Because they don't get into brain parenchyma. And several of these here appear to be associated with olfaction. And the question is, why do we see them upregulated? I mean, it's known, or we could show that the monoclonals go into the nasal lining fluid. So they are excreted and they can work there. So maybe this works indirectly through the olfactory nerves. So, and the pathways, we only have 12 that were positively enriched and 2 negatively enriched ones. But the funny thing is they seem to all deal with detection perception of a stimulus and something with metabolic processes and sensory perception like smell. So something, it does something, but we need to have a closer look. And then we looked at the delta-infected Ronapreve treated versus the mock-infected Ronapreve treated. And all the ones that we saw up compared to the untreated animals were down now. So they're down-regulated compared to the Ronapreve-only treated animals, as if the delta infection sort of counteracts that. But of course, that's very non-scientifically speaking at the moment and needs further investigation. And the significantly affected pathways, I mean, who's surprised? There's still some evidence of an effect on B cell function, but really minimal effects towards innate immune response. So then we looked at the delta-infected Ronapreve treated versus MOC-infected to see whether there is something happening, and that's really minimal. So there are 4 upregulated ones and 1 downregulated, and that's the question: is it due to the small amount of viral RNA in the brain, or is it due to inflammatory processes in the lung? And then secondary, so among them is a protein that's a regulator of macrophage metabolic function. And then one that has to do with ciliary beating. And then there is this one here that's downregulated and involved in the early process of neural differentiation. Others, I didn't find anything about. So we definitely have to have a closer look into that. But now, how could the monoclonal antibodies work? So we apply them intraperitoneally, but it has also been shown in primates and in human patients that if you apply them intravascularly IV, the same happens, of course. They are in the serum and are released into the nasal lining fluid. And there have been other studies using other monoclonals. They come to the same result. We're reaching just the 1 to maximally 2% level of the serum. There is no evidence of tissue penetration, how I would call it, because tissue penetration for pharmacologists is you take the tissue and then you look, but that doesn't mean it goes into the parenchyma, in particular in the brain. I mean, there are vessels in there, so you may just detect anything in the serum. So there's no evidence of access to the brain parenchyma. So we know that the virus, okay, it infects the olfactory epithelial cells, and then you can just about see, yeah, won't be a minute, that it goes into the nerve and via that olfactory bulb and spread. So maybe it actually reduces by reducing the viral load in the nasal lining fluid and then in the olfactory epithelium, we have less of replication, so just less of a chance of neurons to become infected. So conclusion from my side, it's important to understand the cellular distribution of monoclonal antibodies before you draw any other broader conclusion. And determination of tissue antibody concentration in crude tissue homogenates might actually be misleading. So what do the mAbs in the serum do to the brain? So probably something interference with the olfactory system. But we now need to investigate the specific pathways and genes of interest, and we're actually waiting for the final results from our metabolomics, lipidomics analysis. And that's it. Thank you.
Do you have some time for some questions? Louise?
Thanks for it, really interesting. Can we bypass the nasal route with SARS-2 by direct injection into the brain to see what that would do?
I cannot recall that anyone has done it.
Okay.
And I also think it would just make the model even less applicable.
Yeah. No, I was just wondering to see what if you still got the same effect.
I mean, a colleague a collaborator in Helsinki, he has worked with isolating neurons, and he can infect them. It replicates the virus, but when you keep them in culture, or also when you use the brain organoids, they die eventually, but for several reasons. So it's another, I mean, none of these models, let it be in vitro or like this one, is perfect, but we know it can get into the neurons. I think it's a very, very limited infection. And therefore, because our consortium works on long COVID and all the CNS symptoms or subtle changes, so we think there may be an infection to some extent. It triggers something, and then for some reason, this trigger is gone. But the effect remains. So there is, we have another talk tomorrow that also shows that there are some transcriptomic changes that affect behavior. So there's probably something happening. Our model is far too crude. Yeah.
Anyone else? I was just wondering, are the monoclonals all IgG, and are you investigating isotype switching of the monoclonals at all?
So we haven't. I mean, these are, we just looked it up again yesterday. It's a kappa and lambda combination, and they should stay as they were. But what we know is that the human antibodies work in the mouse, so they work fully. And in the prior study, we could actually show that they induce a bit of a granulomatous reaction in the lungs. They seem the macrophages seem to take up the virus and then really bind it, Fc receptor probably, and then it stays there, and it's confined that way. And that could be happening in human patients as well, of course. Okay. Thank you.
All right, thank you.
Yep.